Today's study tip is to use your professors as resources. So come talk to me, ask me questions, go to office hours, send me an email, connect with me outside of these videos so that I know what you have questions about and how to help you. Now we're talking about Chapter 17, the urinary system. This will be divided into two parts. This is the first video of the urinary system. And first we'll talk about functions of kidneys. So the first one that everybody knows about is that the kidneys form urine, but the kidneys also have lots of other important jobs. One is to filter blood, and it regulates a whole lot of things. So it regulates blood volume, ions, pH, and more. The kidneys eliminate waste. These are going to be nitrogenous waste, toxins, drug, and hydrogen ion. And kidneys are in control of acid-base balance. And so we'll get to this in a later chapter when we talk about this. So remind you of the gross anatomy of the kidney. So you have this cortex on the outside, the renal cortex. Surrounding these renal medulla, that's made of these medullary pyramids, they're called. So there's several renal pyramids that make up the renal medulla. And those are going to empty into these minor and major calyxes or calyces that will eventually, the urine will collect here and collect in the renal pelvis and then go out the ureter to the bladder. Okay. The nephron, where the urine is actually made, is going to be lined up all along this cortex. And so we'll see some closer up views in a few minutes here. Okay. We call urine filtrate while it's inside these nephrons. Okay, so here's what a renal pyramid looks like. When you go up closer, here's the renal cortex portion, and here's the renal medulla portion. And you'll see that the nephrons, the functional units of the kidneys, are partially up here in the cortex, and then some of the tubules dip down into this renal medulla. Okay, so we're going to talk about this in detail, but look here what's happening here. Here's the blood flow coming in, and it's forming this little ball of capillaries called the glomerulus. Okay, and the glomerulus is surrounded by this capsule called the Bowman's capsule, which is where filtrate will collect and then will eventually filter through the system. So other things to note here is that we have two different kinds of nephrons. We have cortical nephrons that are mostly up here in the renal cortex. And then we have these juxtamedullary nephrons that dip down way deep into the medulla. So one single nephron tubule looks like this. This is going to be one of your functional units of the kidney. You've got about 1 million of them per kidney. Okay. And so we said it starts off with that Bowman's capsule surrounding the glomerulus, surrounding that nest of capillaries here. And then that filtrate is going to pass from the Bowman's capsule through this proximal convoluted tubule, proximal because it's closed and convoluted because it's twisty. Then the filtrate comes down the loop of Henle, up the other side, through the distal convoluted tubule, and finally out the collecting duct. The process of urination is called micturition, and it's actually under a reflex. We have a micturition reflex. There's a pretty great video here you can watch about this. And this has to do with bladder stretch. So bladder stretch receptors are activated between 250 and 400 milliliters of liquid in your bladder. And the parasympathetic nervous system reflex then contracts this bladder. Okay. So you have urinary sphincters that are made of internally smooth muscle and externally skeletal muscle. So that means that the smooth muscle one is not under your control, right? The smooth muscle one is going to be under autonomic control, and then the external skeletal muscle one is under control. And so both of these then need to relax in order for urine to come out. The opposite of the micturition reflex is called the guarding reflex, which allows the bladder to continue to fill by inhibiting the micturition center in the pons. So something that nurses are going to be using a lot because you're trying to prevent yourself from peeing so you can serve your patient, right? Okay. So there's a whole lot of clinical implications of kidneys and kidney function. One is going to be kidney stones. So about 80% of kidney stones are made of calcium oxalate. They get stuck in the ureters and it can be very painful. So these can cause backup of urine into the kidneys. It can cause inflammation in the area, and it's certainly very painful to essentially pee out rocks. They can be treated, though, with sonic waves or surgery. They can sometimes be prevented with lots of water, though lots of people are what they call stone formers. So their kidneys' anatomy and physiology just makes them more likely to form stones. Incontinence is another clinical thing that is very concerning, which is when the bladder is unable to hold urine. This can change with age and also with things like pregnancy. So pregnancy hormones, and body changes that happen during pregnancy can make incontinence a problem post-birth. Why this happens with age is that aging decreases the strength of sphincters, but there are some things that can help with this. So certainly if you're on the younger side of things like meds, exercise, and time bathroom breaks can help. But it can be really difficult to restore the strength of those sphincters, and so it can be more difficult as one person ages. Okay, so we have 3 major processes to talk about when we talk about making urine. Okay, so we're going to start here at this glomerulus inside the Bowman's capsule. So when stuff goes from the glomerulus to the Bowman's capsule, that's called filtration. Okay, so filtration is the first thing that happens where you put compounds from the blood into the filtrate. That's filtration. And then along all these tubules, reabsorption happens. So reabsorption is when you take molecules from the filtrate back into the blood. So you're reabsorbing them. Finally, we can also secrete other stuff. So we can move more things from the surrounding capillaries into the filtrate. That's called secretion. So filtration is the initial formation of the filtrate. Reabsorption is bringing things back into your blood. Secretion is putting things from the blood into the filtrate. And then finally, you have excretion, which is taking the urine out of your body. Okay. So first we'll talk about filtration. Filtration is moving those substances from the glomerulus to that glomerular capsule, or the Bowman's capsule. We make about 180 liters of filtrate produced per day. Okay. And this gives us a glomerular filtration rate, GFR, of about 125 milliliters per minute is going to be the average or the most typical. At this time, about 20% of plasma moves into the capsule. So really you're pushing a whole lot of your blood liquid then into the filtrate. Okay. So, but does that mean you're peeing that all out? Are you peeing out 20% of your plasma? And the short answer is no. Okay, so how much urine do you actually make per day? We'll think about that kind of question. So once you've pushed all that blood volume, all that plasma into the filtrate, then reabsorption happens. This is, again, moving substances from the renal tubing back into the blood. This is how we save stuff. So we pushed a bunch of things into the urine, and now we want to bring stuff back in. So we're going to save the things that we want to save. This is going to be a lot, 178.5 liters reabsorbed per day. So we're actually reabsorbing most of the stuff that you initially put into filtrate. Okay. Most is going to be things like water, but we'll talk about other stuff too. Okay. So the third process, secretion, is selective removal of substances from the blood to the renal tubules. So if you want to dump stuff out, you can get rid of things in the urine, but this requires your body to recognize what you need and how to get rid of those substances. So it's really actually only a very specific number of molecules that you can do this with. And excretion is elimination through urine. Okay. All right, so first thing, let's look at this renal filtration membrane. This is a really special membrane happening in the kidney. And again, you see the blood flowing in this afferent arteriole to the glomerulus, okay? And blood eventually flowing out of that glomerulus. Then we have that filtration happening from the blood into the filtrate, into the urinary system, through this special membrane with podocytes. So the podocytes line this membrane. There's little tiny holes called fenestra. Fenestra just means window. That is going to be where the plasma is pushed, okay? And so this is actually a really delicate membrane system, starting with the fenestrae in the capillary endothelium here on the bottom. You have that on top of a glomerular basement membrane, and then the podocyte foot processes. And so this is 3 layers to filter through from the glomerulus, which means that a lot of things are going to stay in your blood, right? And we'll talk about that in a second, what those things are. But a lot of stuff's going to stay in your blood. Only certain compounds and things are going to come through that membrane. So the integrity of this membrane is really important, because if you start to make big old holes in this membrane, now you're going to lose things like blood cells into your urine, which you don't want to do. Okay. So the filtration membrane is made up of those fenestrae of the glomerulus capillary, a basement membrane, and the visceral layer of the Bowman's capsule. And this is made of those podocytes with spaces called filtration slits. So to make it from your blood into your urine, it has to go through these 3 parts of this filtration membrane. So what makes it through? Water, salts, glucose, small peptides, but not cells. Cells are too big, and so are big polypeptides and proteins. So cells, polypeptides, and proteins stay in your blood. Pretty much everything else gets filtered out. Once the fluid that is in the urinary system, in the capsule, it's called filtrate. Okay. So now we got to think a little bit about this pressure idea. And we came up with this for the circulatory system, right, where pressure is going to affect blood flow. And here we're going to see that glomerular blood pressure is going to favor filtration. Okay. Glomerular colloid osmotic pressure, though, is going to oppose filtration. What you'll see so far is that if we compare this glomerular blood pressure with the glomerular colloid osmotic pressure, now we have still a net filtration. Right? Capsular hydrostatic pressure, though, also opposes filtration. But neither of those two opposing filtration effects from the glomerular colloid osmotic pressure or the capsular hydrostatic pressure are enough to oppose it. So we have about 10 millimeters that is going to be the net filtration pressure favoring filtration. Okay. So that glomerular blood pressure then is going to be the main driver of, or the primary driver, the entire driver, favoring filtration. And so if we adjust the glomerular blood pressure, then we can also adjust filtration. Okay, so how do we calculate this? Basically, you subtract all the ins from the outs. Just memorize the normal, so then you can plug in any numbers that I give you. Okay, so the one I just gave you there's our example for normal. 55 out minus 30 in minus 15 in is 10 out. Normal filtration. Let's change it up. So in someone with kidney stones, okay, they're going to have an increase in the capsular hydrostatic pressure. Okay, increasing that capsular hydrostatic pressure from 15 to 20. When we do out that, then now we have 55 minus 30 minus 20, so we're only going 5 out, which is still filtering, but it's decreased filtration. Okay. Someone has liver damage, you're going to decrease the colloid osmotic pressure. And so we're going to have increased filtration then, because you're taking away less. Example 4 is a person with low glomerular blood pressure, and so they're going to have a decreased filtration. Okay. So different ways in which we can modify this, but I want you to memorize this normal one so we can talk then about, I can plug in any numbers I give you for any kind of pathology like this. Okay, so regulating this GFR, the glomerular filtration rate, is really important. And we have a lot of different kinds of controls. So the first one we'll talk about is the extrinsic control. The sympathetic nervous system constricts the afferent arteriole. So it's changing this GFR by changing blood pressure. Okay. How is it changing it? Well, if you constrict the afferent arteriole, so you constrict the amount of blood going into the kidney then, GFR will decrease. An intrinsic control is controlled by blood pressure. So if blood pressure drops, the afferent arteriole is going to vasodilate to keep your glomerular blood pressure normal. If your systemic blood pressure is high, the afferent arteriole vasoconstricts to keep this normal. So even though you're going to have changes in your blood pressure throughout the day, over minutes, whatever, your kidneys are going to vasodilate and constrict that afferent arteriole to keep GFR pretty constant. Okay. So now, a question. Explain what would happen to glomerular filtration rate, GFR, and urine volume in the following patients. Okay, so first is someone with liver disease. And I want you to think about how the liver affects albumin levels and then what albumin might be doing. Remember, albumin's really, really, really big. That should give you a hint. Second's a patient with kidney stones. We talked about that a little bit when we talked about that calculation. So think about how the stones are affecting renal tubules and what that would mean to someone's GFR and their urine volume. Okay. Once you've thought about those, come talk to me. We can talk through the answers. Okay. So that was filtration. And we can manage filtration and regulate filtration mostly through that afferent arteriole. So by controlling blood flow to those kidneys. Okay. The next process is reabsorption. A really important thing that's reabsorbed from the filtrate back into the blood is sodium. This happens mostly in the PCT, the proximal convoluted tubule. And it works through a high to low sodium glucose co-transport across the apical membrane. Okay. About 80% of kidney energy is used for active transport across the basolateral membrane. So remember the sodium potassium pump? We had them in the brain, now we have them here in the kidneys. Okay. And actually about 65% of sodium is reabsorbed in the proximal convoluted tubule, a further 25% in the loop of Henle, and only about 10% in the DCT, the distal convoluted tubule. But this one's easy to adjust, actually. Okay, here's what this looks like. When I say the apical and the basolateral membrane, I'm talking about this. The apical side is in the lumen of the tubule. The basolateral membrane is the one that's towards the blood. Okay, so here you can see that sodium is being co-transported from the apical membrane with glucose, right? Because remember, we're reabsorbing, so we're going from filtrate back into blood. Okay, so sodium and glucose is co-transported, and that then creates the ability for the sodium then to go into the blood. Okay, but for this to work, that co-transport to work, passive diffusion, we have to have low levels of sodium in that space between the basolateral membrane and the blood supply. So that's achieved by active transport. Okay, so the sodium potassium pumps, the active pump, is pumping sodium from the basolateral membrane into the blood. That creates a low concentration of sodium then in that space. And now sodium can travel down its concentration gradient from high in the filtrate to low in that basolateral membrane. So this active transporter going from basolateral membrane to blood is what gives us the concentration gradient needed to run this passive diffusion co-transport of sodium and glucose. Other ions and molecules are reabsorbed as well. Potassium, for example, is pumped into tubule cells. We just talked about glucose. Glucose is co-transported with sodium from the lumen to the tubule cell. And this is going to give you 100% reabsorption unless there's diabetes. So with diabetes, things are going to be all out of whack. Chloride follows sodium and potassium passively. So if we're pumping potassium back into the tubule cells, and we have that sodium reabsorption mechanism, the chloride's going to follow. And then water follows the salts by osmosis. So now we have a way to get potassium, sodium, chloride, glucose, and water reabsorbed. An average human drinks about 16,000 gallons of water in a lifetime. There's approximately 80 gallons in a bathtub, so we drink a whole lot of water. In the loop of Henle, we have something pretty cool happening. It's a countercurrent multiplier system, and this helps to concentrate the filtrate further. So we reabsorbed a bunch of stuff in the PCT, and then the filtrate enters the loop of Henle. So we're going to concentrate the filtrate in through this descending limb, and water leaves via osmosis while salt stays. Okay? Then it gets diluted a little bit through the ascending limb when salt leaves. So this gives us a great amount of reabsorption power for both salts and water, because water is going to be reabsorbed in the descending limb, and salt is reabsorbed then in the ascending limb of this loop of Henle. Okay? Here's what it looks like. And what you can see then is once you have filtrate coming in down this descending limb, you can see it increase then in milliosmolarity as you get down to the bottom. And that's happening as water is reabsorbed. But then in the ascending limb, now you have salt being brought back in. And so that is going to decrease the milliosmolarity, but you've already brought back in a bunch of water. So you're also losing volume here, okay? And now, losing meaning you're bringing it back into your blood. You're actually keeping that volume, but losing urine volume. So by the time you pee that out, you're going to have a much lower, more concentrated volume of urine. Okay. So as a reminder, the descending limb is impermeable to salts, but permeable to water. The ascending limb is impermeable to water, permeable to salts. So that means that we can use active pumps to pump those salts back in, reabsorb those salts, and then water is going to follow through osmosis. Okay, so we don't need active pumps for that water permeability. Okay, So it's putting it all together, showing you mostly that this urine is closely next to the bloodstream. So you see this glomerulus coming in, but you also see this loop of the bloodstream coming down too to be right nearby. So it can reabsorb anything that you're reabsorbing into your body then. Okay. So finally, we're going to talk about antidiuretic hormone. Remember, this came up in a blood pressure chapter. So antidiuretic hormone, its job is to increase permeability of collecting duct to water by stimulating an aquaporin channel. This allows you to increase water reabsorption and can really regulate then urine concentration. So urine concentration varies wildly from 100 to 1400 milliosmoles. Okay. And if you increase water reabsorption then, that causes an increase in blood volume, which increases blood pressure. And now you're like, oh yeah, ADH increases blood pressure. So when is it stimulated? It's normally stimulated when you have low blood pressure. Tells you to reabsorb more water from the kidney in this mechanism. That increases blood volume, and then that increases blood pressure. So it helps you save water. That's why it's called antidiuretic hormone, because antidiuresis makes you pee less. This release is controlled by the hypothalamus and osmoreceptors. ADH is also inhibited by alcohol. So drinking alcohol inhibits ADH secretion, although this exact mechanism is unknown, which is pretty interesting. So I want you to think about then, how would drinking affect blood volume and blood pressure? Think about that. Come back and talk to me. And then in part 2, we'll move into some more specifics about the urinary system. Okay. You can talk about also with me about how or why, if ADH secretion is reduced, what's happening in the kidney. So think back to those mechanisms and about aquaporins. Okay? And I will see you all next time.